Hello, my name is Zdena Jandarova, and I am presenting my research poster on hoverfly movement toward natural, full-spectrum artificial, and specific wavelengths of light, specifically looking at the species Eristalis tenax, which are native to Georgia. I conducted my research project under the guidance of Dr. Jason D. Lang from the Biology Department at the University of North Georgia in Gainesville, Georgia. Research groups study hoverflies and use them to observe hoverfly behaviors such as movement, pollination, and feeding. Hoverflies are positively phototactic species, moving from dark to light. This behavior is manipulated by research teams and is most often used to transfer hoverflies from soiled cages to clean cages. Our research team observed a difference in the rate of phototaxis between two different locations. Originally, a hoverflies were placed in a greenhouse exposed to natural light and temperatures and a laboratory exposed to artificial light and room temperature. We noticed hoverflies were more active under natural light in the greenhouse compared to artificial light in the lab room. Thus, we had two hypotheses. First, we hypothesized that the greatest percentage of movement would occur under natural light. Secondly, we hypothesized hoverflies would move sooner under natural light compared to non-natural light conditions. Our research team tested movement by covering the hoverfly occupied cage with a blackout curtain and attaching a clean, unoccupied cage exposed to different types of light to allow the hoverflies to move independently. The different types of light we used were natural, full-spectrum artificial, and purple, blue, green, and red LED lights. We ran two control trials, one with both cages covered with blackout curtains and the other with both cages left uncovered under artificial light. Our team ran 30-minute trials for each of our 8 experiments with 4 replicates each. When looking at the percent of hoverfly movement, our research team found that natural light had the greatest positive effect on hoverfly movement compared to the artificial light in the other light trials as well. Under artificial fluorescent lighting and purple LED lighting, the percentage of hoverflies that transferred cages was approximately the same. When comparing hoverfly response to light wavelengths across the visible spectrum, the highest percentage of hoverflies moved toward the short wavelength spectrum or towards purple and blue light. We detected no movement under red and green LED lights. When looking at the timing of hoverfly movement, we found that hoverflies moved earliest under natural light. Under natural light, the percent of hoverflies that moved in the first 5 minutes of observation was equivalent to the percent of hoverflies that moved in each of the other light trials. The data we collected supports our hypotheses. Our flies moved in greatest proportion and sooner under natural light conditions. Our data shows that natural light works best for moving hoverflies followed by artificial and purple light. Future experiments that could be done with our project include testing the effects of an LED light bulb that resembles natural light on hoverfly movement, testing the effect of temperature on movement, and looking for effects of different colored light on feeding and/or pollination behaviors. In conclusion, researchers could use these findings to help them decide on the best lighting for cage transfer or behavior manipulation, depending on the resources available and most accessible to them.